Assalamualaikum warahmatullahi wabarakatuh. We are from group 6. We'll present about the mucosal immunity. So this is our group that consists of 6 members.
Question number 1 is the introduction to the gut microbiota and microbiome. The gut microbiota is a vast and complex collection of microorganisms that colonizing the gastrointestinal and profoundly affect human health. Previously, people referred to the gut microbiota as microflora of the gut. The microorganisms that live within the gut is completely unique to each individual. The human's gut microbiota consists of over 100 trillion microorganisms and weighs around 200 grams. The gut microbiota has 4 main functions, which are promotes digestion by facilitating the absorption of nutrients, helps the digestive tract to function properly, creates barrier against pathogens and toxins, and lastly plays a defensive role in the development of the immune system. The microbiomes is the community of microorganisms that exist in a particular environment. In humans, the term is often used to describe the microorganisms that live in or on a particular parts of the body such as the skin or gastrointestinal tract. So for humans, bacteria that live in the guts help in the digestion of food, and bacteria that live on the skin help to break down the lipids to produce natural moisturizing factor for skin. This group of microorganisms are dynamic and change in response to a host of environmental factors, such as exercise, diet, medications, and other exposures. Most of the microbiomes are symbiotic, where both of the human body and microbiota benefit, and some, in smaller numbers, are pathogenic that promoting disease. In a healthy body, pathogenic and symbiotic microbiota coexist without any problems. But if there is a disturbance in that balance that brought on by infectious illnesses, certain diets, or the prolonged use of antibiotic or other bacteria, it will stop the normal interactions. As a result, the body may become more susceptible to disease.
Question number 2. The crosstalk between gut microbiota and the immune system. The human gut is an ecosystem consisting of a great number of commensal bacteria living in symbiosis with the host. Dynamic interactions between gut microbiota and a host innate and adaptive immune systems play key roles in maintaining intestinal homeostasis and inhibiting inflammation. The gut microbiota metabolizes proteins and complex carbohydrates, synthesize vitamins, and produce an enormous number of metabolic products that can mediate crosstalk between gut epithelial and immune cells. Gut-associated lymphoid tissues, GALs, are a part of the mucosa-associated lymphoid tissues, MALTs, that act as the front line of gut mucosal defense, which lining directly between host and environment. The histological components of GALs mainly includes biopatches, crypt patches, isolated lymphoid follicles, appendix, and mesenteric lymph nodes. These GALs recognizing pathogens by initiating the innate immune response and presenting antigens to activate the downstream adaptive immune system. Through the detection of invading pathogens and the blocking of their escape from the gut lumen into circulation, intestinal immune cells create tolerance toward the commensal microorganism. The dual function of GALs is critical to homeostasis between gut microbiota and human immune system. Next is the advantages of the relationship between gut microbiota and the immune system. First advantage is development of immune system, where the gut microbiota helps shape the development and maturation of the immune system, particularly during early life. Exposure to various microbial species stimulates the immune system, aiding in the development of immune cells and promoting immune tolerance. Next is antimicrobial defense, where the gut microbiota provides colonization resistance against potential pathogens by competing for resources and producing antimicrobial substance. Beneficial bacteria can produce antimicrobial peptides and metabolites that inhibit the growth and colonization of harmful bacteria, protecting against infection. The third advantage is barrier function, where the gut microbiota helps maintain the integrity of the intestinal barrier, which is crucial for preventing the entry of harmful pathogens into the bloodstream and tissues. After that is the host microbe mutualism. While the immune system helps control the gut microbiota by eliminating pathogens, it also recognizes beneficial microorganisms and establishes a mutualistic relationship. The immune system selectively tolerates and supports the growth of beneficial commensal bacteria, which in turn contribute to immune system regulation. The last advantage is immunomodulatory response, where the gut microbiota can produce various such as metabolites, lipopolysaccharides, and specific bacterial components that interact with the host immune system. These interactions can modulate immune system, cytokine production, and overall immune response. This modulation helps to maintain immune homeostasis, preventing excessive inflammation and promoting immune tolerance.
Question 3. Why IgA is important for gut immunity? The first line of defense in mucosal secretion is circulatory IgA. Numerous and diverse microbial communities colonize the gastrointestinal tract. The gastrointestinal system contains about 80% of humans' body antibody secreting plasma cells. They produce circulatory IgA in mucosal protection to prevent the invasion of pathogenic microbes and penetration of intestinal barrier. Secretory IgA facilitate the elimination of antigen and pathogenic microorganisms from the intestinal lumen by a process known as immunoexclusion by preventing their access to epithelial receptors and trapping them in mucus and promoting their removal through peristaltic and mucociliary process. Secretory IgA can also prevent bacterial growth and virulence to the gut epithelium. Secretory IgA-driven clumping via either ancient growth or classical agglutination that leads to the inhibition of virulence mechanism that require contact with gut epithelial cells. Based on this figure, IgA containing the joining J chains that produce as an antibody dimer by plasma cells bind to the polymeric immunoglobulin receptors, PIGR, leads to transcellular transport of IgA to the apical surface, where PIGR is proteolytically cleaved to form secretory IgA. A. When secretory IgA is released, it immediately binds to the luminal bacteria in the antigen-specific valve. B. It has been demonstrated that intestinal bacterial species that have been significantly coated with secretory IgA exhibit cholitogenic characteristics in a disease setting. C. Secretory IgA-mediated clumping of ancient daughter cells control invasive pathogenic bacteria by preventing their separation after division and allows en bloc clearance.
Question number 4. What is the impact of the gut microbiota on health and diseases? The composition of gut microbiota differs between healthy and pathological situations. Eubiosis is as an example of an interspecies balance of the microbiota community, whereas dysbiosis is a disruption of eubiosis. Dysbiosis is frequently defined as an imbalance in the gut microbiota community related to disease. This imbalance could be caused by changes in the relative abundance of microorganisms, which can lead to the infectious and non-infectious disorder. First is about gut microbiota towards the human metabolism. The biochemical profile of the diet is influenced by the gut bacteria, which in turn will influence the host health. The human gut microbiota also contributes to the fermentation and anaerobic breakdown of the food fibers, proteins, and peptides. Bacteria species, particularly those of the phyla Bacteroidetes and Firmicutes, can ferment fibers to create branched short-chain fatty acids, lactate, ethanol, hydrogen, and carbon dioxide, which are either utilized or eliminated by the host. Gut microbiota also can synthesize essential vitamins including biotin, folate, and vitamin K, and also has ability to neutralize carcinogenic compounds such as pyrolysates. Next is about gut microbiota towards the malnutrition and fasting. The disruption of the circadian physiological rhythm increases the risk of intestinal dysbiosis, such as diabetes, intestinal inflammatory disorders, and even cancer. Intermittent fasting (IF) helps to modulate gut flora by enabling continuing interaction with digestible foods and altering intestinal immune responses during the development of chronic heart disease, blood pressure, and diabetes mellitus. The leading causes of child mortality is malnutrition, must be treated with nutrient-rich therapeutic foods. However, they cannot completely recover from body mass improvement, probably due to their immature microbiomes. Gut microbiota towards cardiovascular diseases. Gut microbiota is involved in the metabolism of choline, phosphatidylcholine, and carnitine, which eventually produce trimethylamine and oxide, which are also known as TMAO. TMAO is suggested to not only regulate cholesterol balance and bile acid levels, but is also associated with early atherosclerosis and high long-term mortality risk of CVDs. Short-chain fatty acids, particularly propionates and butyrates, were shown to protect the host from hypertensive cardiovascular damage. Propionates is suggested to regulate the balance of fatty cells and regulatory T cells, which is critically important in hypertension and hypertension-induced organ damage. Lastly, gut microbiota towards cancer. The gut microbiome has also been linked to the development of colorectal cancer, with Bacteria fragilis, Fusobacterium nucleatum, and Streptococcus anaerobic being potential players. While Helicobacter pylori is a known risk factor for gastric cancer, new research reveals that Clostridium and Fusobacterium bacteria are also higher in people with gastric cancer. According to recent research, Fusobacterium nucleatum can reduce the host immunological response and boost cellular growth. Therefore, a diet rich in whole grains and dietary fiber, on the other hand, is associated with a decreased incidence of Fusobacterium nucleatum positive cancer, showing that the gut microbiota may be significant link between dietary and colorectal cancer relationships.
Question 5. Explain gut and brain axis.
For my part, I will explain about the gut-brain axis, or can be called GBA. So, what is GBA? GBA involves the bidirectional communication that connects our enteric nervous system, or our gut, and central nervous system, which is our brain. The communication between these two systems is connected by vagus nerve. Vagus nerve is one of the biggest nerves connecting our gut and brain. This nerve sends signals in both directions and will allow the flow of communication between these two systems. For linking the communication, it is through chemical messengers known as neurotransmitters produced by gut microbes used in the brain for various functions, such as regulations of hormones. GBA linking emotional and cognitive centers of the brain with peripheral intestinal functions. Based on this picture, we can see the involvement between these two communications. So, for example, from gut to brain, as I already said, it can help in regulation of mood. Firstly, our microbe in the gut will produce one of neurotransmitters, which is gamma-aminobutyric acid, or GABA, which can control our feeling of fear and anxiety. Then, from brain to gut, have you ever felt intestinal pain during times of stress? This is because the brain will influence our gut, which is disrupting gut motility and stomach squeeze, then move waste through the body when we feel stress. Both examples have shown the brain and gut have interaction that involve in response to physiological and behavioral triggers. For more detail on how both systems work in many pathways, the next presenter will explain about it. Thank you.
Question 6. How does gut-brain axis work?
Brain-gut axis work in four major pathways, which are neurologic pathway, endocrine pathway, metabolic pathway, and immune pathway. In neurologic pathway, it involves vagus nerve, the enteric nervous system, and the activity of neuron transmitter within the gastrointestinal tract. Neurologic modulation of afferent sensory nerve directly produce molecules that can act as a local neurotransmitter such as GABA, serotonin, melatonin, and acetylcholine. The autonomic nervous system also influences immune system activation in the gut, for example, by directly modulating macrophage and mast cell response to the luminal bacteria. Next, in immunologic pathway, the gut microbiome influences the inflammation metabolism within the GI tract via the release of cytokines and communication mediators such as interferon gamma during times of dysbiosis. For example, in irritable bowel syndrome IBS, abnormal microbiota population activate mucosal innate immune response, which increase gut epithelial permeability, activate gut pain sensory pathway, and dysregulate the enteric nervous system. In endocrine pathway, gut microbiota alters nutrient availability and thus influence the release of biologically active peptide from enteroendocrine cells. For example, the neuropeptide galanin is thought to be involved in many important neurobiological functions such as neurotrophic function, sleep-wake cycle regulation, blood pressure regulation, and parental behavior. Galanin also stimulates the activity of the central branch of the hypothalamic pituitary adrenal and thus enhance the glucocorticoid secretion from the adrenal cortex. Last but not least, in metabolic pathway, bacterial metabolites short-chain fatty acid SCFA, which are produced by the bacterial fermentation of dietary carbohydrate, are decisive humour influencer. It is best known to affect the nutrition of enterocytes, exert significant hormone-like activity, have immunomodulatory properties, and interact with the nerve cell by stimulating the sympathetic branch of autonomic nervous system. Furthermore, microbiota-derived SCFA are able to cross the blood-brain barrier and regulating microglial homeostasis, which is required for proper brain development and brain tissue homeostasis.
This is our references. Thank you for watching our presentation.